Well, hello everyone. I'm delighted to have Rachelle here on the call. First and foremost, just want to say thank you, Rachelle, for joining us. She is the principal scientist of Novo Nordisk, and I just thought it would be a great opportunity to learn more about what you're currently doing right now, and also learn more about the upcoming Event 3D Tissue Model Summit taking place in April. So yeah, first and foremost, just want to say thank you, Rachelle, for joining us, and I'm looking forward to learning more about what you're currently doing and how you're contributing to the space.
Great. I'm happy to be here. Thanks, Michael.
Nice, nice. Well, let's kick us off with the first question. Can you tell us a bit more about your background in regards to these complex in vitro models?
Yeah. So I span almost 20 years, mostly before it was ever called complex in vitro models or microphysiological systems or organs on chips. I started my career, I got a PhD in biomedical engineering, and I started my career at Pfizer, where I was helping to build these primary human-based cell models to model skeletal muscle and liver. And we were supposed to be testing exploratory targets for metabolic disease, mostly diabetes and insulin resistance, but also looking at fatty liver targets as well as hepatosteatosis. Then I worked at Draper, where we worked on a DARPA program, and we were developing a human upper airway model where we were looking at selective pressures and how that induces influenza viral evolution. So looking at things like oseltamivir, small molecules, breathing, and such. And then from there, I moved on to the VIS, where I worked for 6 years, and I helped on Ingber build the organs on chips team and using them for mostly disease modeling and therapeutics discovery. Realizing that disease modeling was really probably a big way for us to start looking at different drugs and how they treated those modalities. And then also managed the last 3 years of the DARPA MPS program, and the goal was to link 10 organs on engineered platform to predict therapeutic efficacy and toxicity. And then I also played a large role in helping to build the Vaginal Microbiome Consortium that was funded by the Bill and Melinda Gates Foundation. We developed the first vagina on chip, which was a nice big step forward in women's health. So that was very exciting to be a part of. And then finally, I also am full circle now. I have been at Novo Nordisk for 2.5 years. And here I am mostly focused on looking at liver models and how we can utilize them to understand the organ's role in diabetes and insulin resistance, as well as MASH and fibrosis. And we are currently using them more at the very early drug discovery phase of target discovery. So
nice. Thank you for sharing that. There's a lot of experience that you've got there. So yeah, really looking forward to learning more about your experience as well. But before we get into it, I'm curious, what were complex in vitro models called before they were called complex in vitro models?
So we called them, it was first called it, so when I first got hired, it was called the Tissue Engineering Group because it was called Tissue Engineering. And some of the first people to actually think of, oh, maybe we could use these models as a translational model for drug discovery, efficacy, toxicology type work were Linda Griffith. Don Ingber was obviously also a big person in this, as well as Mike Schuler. So they were people from the late George Whiteside. So I actually have a timeline I can share that I had put together for Novo to kind of just say this is not really new. These ideas have been thought. It's been the technology that's had to come along and really help us move forward. So yeah, but we were called the Tissue Engineering Group, and that was the key word or buzzword when looking at 3D modeling of tissues. Yeah.
Oh, nice. Yeah, I've heard of that phrase, tissue engineering and tissue engineering. Yeah. So it's good to know like where it all came from. So that's quite interesting.
I mean, now it's still used tissue engineering, but it's more for actual medical clinical replacement of tissues. So yeah. So
interesting. Well, another thing I wanted to discuss as well was from the conversations I've had as I've been researching this space, is that there is a challenge around applying these more complex in vitro models to larger preclinical projects. So what do you think is needed for complex in vitro models to be used alongside more traditional in vivo models?
First is alignment and a full understanding on both sides of what each brings to the table. We do a lot of, I mean, my whole life has been a lot of communication and understanding, you know, what people are happy with in vivo models, and what they're not happy with, and then how, when you say in vivo models, you're thinking of animal models, like what can't they replicate, and then what are we trying to replace? So, and then the other thing that's very important is to test, like, you know, what is achievable with in vitro models, and then understanding kind of how, because you're always limited by medium conditions or structural, or, you know, maybe materials aren't available. So what can you provide? And I think what a lot of people forget is, what can't you produce? Everybody's always worried about, like, just not achieving that goal, but that also tells you something about a mechanism or stuff. So really kind of understanding that biology, but really having a nice connection with the in vivo model team and understanding what they bring to the table, and then how you compare with those functional endpoints. So
interesting.
Yeah.
Another thing that I wanted to mention as well, you've probably seen this a lot, especially at last year. There's been a lot of news regarding these complex in vitro models, right? You've got, you know, the Senate clearing the FDA Modernization Act, people know. You got the FDA obviously releasing their roadmap as well. You've even got like NIH, and you can see that their stance is to move towards these more complex in vitro models. So with all of that being said, right, how should biopharma respond to all of this news here?
I think that, so biopharma has always had some role. Like, there's a lot of positions out there. I think what biopharma needs to do is really embrace it. You know, they've now come out with more specific guidelines, because, you know, before it was, you know, 1.0, 2.0, and it wasn't as specific, but now they give you an actual problem. I think biopharma really needs to kind of do a little bit more of the heavy lifting and really try to implement the MPS into translational and regulatory efforts. Earlier in my time in this field, like more in like the 2015s and stuff, a lot of biopharma was kind of treating it more of a niche thing. It got put in toxicology, or, you know, and such. But if we can really start pushing translational and regulatory efforts, because I think the only team that can do it is industry, biopharma industry, with the help of providing guidance to the FDA, as well as leveraging a lot of the work that academia has done. So I really hope that we can find that path, and that path won't be the same as it looks the first time in the future, right? We'll have it, it'll be changing, but that's how we're going to learn. So is by implementing this and then seeing what the outcomes are, and then how we retrospectively compare what we saw. So that's always been the hard thing. So yeah.
With all of the news, is there still a sense of hesitancy when it comes to deploying these models? And if so, like, why is that the case?
I think, so to be fair to industry, and I understand it, is that we need more robustness, is what they'll say, or, you know, and so then how do you model robustness? And a lot of people have, especially the IQ consortium, has done a great job at putting together these papers and stuff to say, this is how you can consider, you know, functional metrics for liver toxicity. And so I think, you know, I think coming up with an actual agreed upon set of what those metrics look like, how they translate from the human, are going to be key into getting initial buy-in. I know many people have been working on it, but I have yet to see a true implementation or buy-in from the FDA, if you will. So hopefully with the onset of this 3.0 Modernization Act, we'll see it opens people's eyes. They're more excited, right? It cuts down on, you know, NHPs are very difficult. Animal studies can be very expensive. So if we can all come together and say, well, this is how we're going to do it, and then each of us can learn from that, it'll be hopefully bought in. But yeah, I think it's more the robustness and getting agreement on what robustness looks like. So yeah.
Interesting. Another question as well. Very broad question, but I want to see where you're thinking here. If you were to solve one challenge regarding, you know, these complex in vitro models, what would that be and why?
They're more, I would say, a little bit more political. I think we need more open-mindedness, and that's not just for the late stage to answer to the Modernization Act or filing IND. But it's also early stage. I think we need more multidisciplinary efforts because we all don't speak the same language, and a lot of these things have never been done before, so the answer or path is not there, and we have to build it. I see that every day at work. But then also, I think on the other side of it, like, you know, we need more to push for more of a clear path. And I think industry is going to be the one to do that. And so it's political, right? Because everybody just wants to use an animal model because they trust it and such. But I'm hoping that with this new Modernization Act, we'll be able to really push the envelopes and build, you know, that path to see if MPS are effective. And I want to say to see if MPS are effective because we really don't know yet. Right. I think there's been a lot of like short communications and stuff saying that we used MPS to deliver this drug to the clinic, but I think it's a bit weak. But here, if we actually can, you know, deliver an antibody therapy, a very targeted mechanism, very low risk. Well, not everything's low risk, but as low risk compared to the others, then I think that will help us. So, but I think we need to be more open-minded. We need to push multidisciplinary efforts. We need to be more objective about our data. And I think once we start doing that, that will help regulatory accept because they're very strict on what they're putting into a human. So how do we get to where the animal models are? And that's going to be a lot of data. That's going to be a lot of organization. That's going to be a lot of communication. So.
And when you're talking about data and a robustness, are you also like referring to things like model validation as well?
That should have been done already, probably.
Okay,
so I'm talking. So yeah, I mean, what goes into? They want to see more than probably 10 donors in the system. They want to see. They want to see more than one positive control. They want to see negative controls, because that's really how we can tell where our actual therapeutic is behaving, because we have a lot of data most likely with those positive and negative controls in the human. So how does it behave and compare to that, as well as in the animal model? It's never going to be just one answer, right? It's going to be a multiple of experiences and data and testing to kind of build confidence in an organized manner. So yeah.
Yeah, no, it makes sense. And going back to the conference, help us understand, like, why is it essential for, you know, people who are so focused on these complex in vitro models to take part in the conversations happening at the 11th 3D Tissue Model Summit?
Well, first of all, there's just been a lot more buzz and more government efforts, not just in the United States, also in Europe, to maybe reduce our use of animal studies. Secondly, it's a smaller meeting, but it's a great meeting to really meet everybody from all walks of life that have touched MPS, industry, academia, and government. And it's really a great place to come together and discuss the gaps, both political and scientific. And then we can also devise solutions. And I also really like to see how people are coming along. You know, what is the interest? For example, I had someone new I had met in a company, a big company, and basically with the onset of the FDA Modernization Act 3.0, it finally got the attention of his higher ups, right? Like, why aren't we doing this? So yeah, I just can't think of a better place to really kind of meet people and be open-minded and talk about experiences and come up with solutions. So.
Nice. And last question for me. What are you most looking forward to at the event?
So last year I went and the Modernization Act 3.0 had just come out. So a lot of people really didn't have a lot of time to discuss it. I'm really looking forward to kind of seeing how everyone's thought process has developed and what experiences they've had in the industry. Like I said, I've been in the field for almost 20 years, and I'm very much looking forward to catch up with old and new colleagues to understand how their experiences have changed or stayed the same in industry and academia and government. And basically, it really just is a great place to kind of see where we are have we moved that needle at all in a year? So.
Nice. Well, thank you, Rachelle, for your time. I really appreciate the insights that you've shared with us today, and particularly looking forward to seeing your contribution with our workshop B, where we'll be tackling the challenges around self-sourcing, but not only that, also your contribution to day 2, where you'll be contributing to the challenges around model reproducibility. So yeah, thank you for your time. And for those who are watching this interview, thank you for watching. And I hope to see you guys there as well. It'd be nice to see some new faces. So until then, I hope you guys have a nice rest of the day. And I'll talk to you guys soon.
All right. Thank you so much, Michael. I really enjoyed this. I'm looking forward to April.
Nice. Me too.
All right.
Me too.
Okay.
All right,
All right.
Take care.
Bye.